Right, Tristan taking a while. Okay. Now this was posted since last week. If you look under cell division, you would see the slide. I have mitosis and meiosis under cell division. All right, so let's go through meiosis. So this photo is actually, the background here is actually a microscope image. This is actual cells and how it looks when it's divided by meiosis. All right, now we have to understand the whole process of it, the importance, and also we are going to compare sexual and asexual reproduction. So first to begin, let's talk about what meiosis is all about. Now it's a different type of cell division. However, the main difference is it occurs in the reproductive organs. So it occurs, the two cells that are going to divide by meiosis are the sperm cell and the egg cells. Those are the only two cells in the humans that reproduce, that forms copies of each other through this process. Now, meiosis, it tends to form four daughter cells. So where you notice my mitosis forming two, this one is formed four because it have two kinds of, it's two divisions are taking place that is going to result in four daughter cells. You'll see it in the figure. Now, each daughter cell is going to contain half the number of chromosomes as the parent cell. So what that is saying is each sperm cell is going to only contain 23 chromosomes, not pairs, chromosomes. The egg cell is going to contain 23 chromosomes. And it's important to have the number because that's the only way in which you're going to get an organism that is going to form when the sperm and the egg meets and fuses together. Now, meiosis tend to have unidentical cells that are formed. Mitosis is identical cells, so that's another difference. All right, now half the number of cells, half the number of chromosomes, sorry, why am I saying cells? Half the number of chromosomes is what you call a haploid number. Haploid meaning is one set of chromosomes, which is 23 chromosomes that's going to reproduce. So why is meiosis important is that it's each daughter cell or gamete has haploid number of chromosomes. So that's important where you have sperm cell have half, egg cell have half, so that when they fuse, when they fertilize, you're going to have a diploid number that can be restored. So when the egg and the sperm fuse together, you get that 23 and 23, 46 chromosomes. And that is the start of the development of a organism. All right, and then the second importance is that each daughter cell or gamete has different combination of genes, which leads to variation among offspring. So in mitosis, we saw that the daughter cells are identical. But in meiosis, it's not identical because if it's sex cells or gametes that are dividing, they each have different combinations so that each offspring that is going to form from the gamete is going to be different. So that is why you are different to your siblings. Each sibling in your family has its unique set of genes or unique combination of genes. So there's variety in the offsprings. All right, no one is identical to each other. Everyone is different. There may be similarities, yes, but they are different. You are two different, unique offspring or organism. And that's important for the survival of the species in the long run. Right, so here we have all these stages. Now this one is long. You have 1, 2, 3, 4, 5, 6, 7, 8. Now in this one, it's two divisions taking place. And they normally put telophase and cytokinesis as one. Some books have it separate, but sometimes they see it together. All right, because right after telophase comes cytokinesis in the events. Now, the first phase, notice they did not start with interphase. They go straight into prophase. So that's a difference you notice. So let's look at what is taking place. Now, the parent cell in prophase 1 is going to have chromosomes that are already in this thick, dense, condensed shape. So remember in interphase, it's seen as threads, but prophase, we already meet it in this condensed shape. But a lot of things is happening. They say prophase 1 is one of the longest phases because a lot is happening in this phase. What we are seeing is, we are seeing centrioles, these little structures, are forming spindle fibers. We are seeing the nuclear envelope or the nuclear membrane is breaking into pieces or fragments. We are seeing the homologous chromosome. Homologous means same chromosome or similar. They come in together, or they are basically crossing over each other. Now, why are they coming together is because you're going to have swapping of genes taking place when they come together. So the blue will indicate the paternal chromosome. The red will indicate the maternal chromosome. So one is from the mother, one is from the father. So when they come close together, they start to overlap with each other, as you see in here. Right? Pieces of each other are going to overlap and it's going to swap. So genes are going to switch from mother to father. Mother to father. So you have gene switching. You call this chiasmata when they actually switch. So a lot is happening in prophase 1. The chromosomes condense and the nuclear envelope breaks down, crossing over occurs. So when the homologous or similar chromosomes come together, they cross over and they swap genes. Later into metaphase, they align themselves along the equator. So you have the both of them, the homologous chromosomes, aligning itself, and then you have your spindle fibers. They call it microtubule as well. That's another name. They attach to the chromosomes onto the centromere region. So the centromere, the spindle fiber is attached to the centromere that's holding it in place. All right, so pairs of homologous chromosome move to the equator of the cell, which is the center of the cell. And in anaphase, they are pulled apart. So now that we have our chromosomes that has exchanged parts of, exchanged genes, so you're seeing bits of red, bits of blue, that had occurred in prophase 1 when they crossed over. So you're seeing pieces of each other, and then they now are going to be separated. So you have one set of chromosomes going on the opposite end and the other going below. So they have been separated and they are moving to the opposite poles. That's anaphase, part 1. Telophase, part 1, is when the cell starts to actually divide. So you have the cleavage that forms. Then you have the nuclear membrane that is forming back, that brown, those little brown dots. That is the nuclear envelope. The nucleus is starting to reform. The centrioles move up opposite. The spindle fibers are totally broken down. So here the cell is going to split into 2. And both of them are going to contain, so you have, in this aspect, you're going to still have that diploid number of chromosomes, and then in the next division is going to half. Right? So here we have the chromosomes, both on the opposites. They have divided. You have 2 cells being formed. Second division, those 2 cells that you get from part 1 are now going to undergo the same process. So again, prophase 2, the chromosomes start to, they are already in that condensed shape. this nuclear membrane breaks down again. You see how it's breaking down in both of them? Spindle fibers start to form, centrioles move apart. Metaphase 2, chromosomes align themselves, both cells. The spindle fiber attaches to the centromere, which is the center of the chromosome. But in anaphase, look at what happens. That chromosome structure that you saw has split into two. So you have two in opposite ends. And then you have two in opposite ends. This is where the chromosome has halved. It has halved itself. So you have half the number going on either sides. All right, and then in the telophase 2, the cell starts to divide. So now that you have half the number on both sides, so you have 23 chromosomes here, 23 here, 23 on this side, and 23 on this side. So each cell, all four, now you have 1, 2, 3, 4 cells. 4 daughter cells containing 23 chromosome, which is half the number of chromosome as this parent cell from prophase 1. All right, so that is how you're going to get your haploid set of chromosomes at telophase 2. All right, and then in nuclear envelope, when it will reform, chromosomes inside of the nucleus, centrioles starts to go back to its original position. That's all that is taking place in meiosis. So it's multiple steps, but basically your parent cell is what has a lot of things happening, such as crossing over, homologous chromosomes pairing up, exchange of genes taking place. This is where the crossing over is what leads to variation. And also when they align themselves along the equator, this could be random. So this chromosome could come here, this one could come there. This is also cause variation to occur. You call it random assortment of chromosomes. All right, so those two features in meiosis allows variation or the offspring that is going to form to be totally different than their sibling. All right, let's look at a video to tie in all of these stages together so you can see it. Okay, might be a bit loud. I'm giving you all the disclaimer. You might want to lower down your volume a bit. Or in some cases, it might be too soft. So I'm going to start playing it. You look at it carefully. I want you all to examine the different stages of meiosis, all right?
Happens during the phases of meiosis. Meiosis, sometimes called reduction division, is the type of cell division that produces gametes. By gametes, we mean sex cells, such as sperm cells in males and egg cells in females. Meiosis is broken down into two stages of cell division, called meiosis I and meiosis II. Meiosis I has 4 phases: prophase I, metaphase I, anaphase I, and telophase I. And meiosis II also has 4 phases: prophase II, metaphase II, anaphase II, and telophase II. Let's look at what happens during meiosis I. Prophase I starts with a diploid cell. Its chromatin contains 2 uncoiled, spread out sets of chromosomes, one from each parent. After the DNA in the chromatin replicates, it condenses into the more familiar X-shaped chromosomes. The replicated DNA is the same in the identical sister chromatids of each chromosome. In a process called synapsis, each chromosome pairs up with and binds to its corresponding homologous chromosome, forming a tetrad. A tetrad is the group of 4 sister chromatids in paired homologous chromosomes. The chromosomes contain genetic information called genes. These genes were inherited from each parent. And different versions of the same gene on each chromosome are called alleles. In a process called crossing over, chromatids from each homologous chromosome exchange segments of alleles. Also called recombination, crossing over randomly happens on every chromosome, resulting in different gene combinations. This explains why every gamete is genetically different from every other gamete. Crossing over results in genetic variety in offspring. This is why children are different from their biological parents, as well as from their biological siblings. Continuing on with prophase 1, the nuclear membrane disappears. The centrioles move to opposite ends of the cell, and spindle fibers fan out from them. Next, in metaphase 1, the homologous chromosomes line up at the equator and attach to spindle fibers from opposite poles. During anaphase 1, spindle fibers separate the homologous chromosomes in each tetrad and pull them to opposite poles of the cell. The cell enters telophase 1 with 1 chromosome from each homologous pair at separate poles. However, each chromosome still consists of sister chromatids. Keep in mind that each chromosome's sister chromatids are no longer identical because of the allele exchange that happened during crossing over. Then, spindle fibers disappear, and the nuclear membrane reforms around the chromosomes. Finally, cytokinesis occurs. Meiosis I ends with 2 genetically different haploid daughter cells. Each haploid cell contains only 1 set of chromosomes consisting of paired sister chromatids. Both cells now enter the next stage, meiosis II. However, unlike meiosis I, DNA does not replicate before meiosis II begins. Once again, in prophase II, the nuclear membrane disappears, and spindle fibers fan out from the 2 sets of paired centrioles. During metaphase II, the chromosomes in each cell line up at the equator and attach to spindle fibers from both poles. During anaphase II, the sister chromatids of each chromosome separate and move to opposite poles. Once the sister chromatids separate, they're called chromosomes. Finally, during telophase II, the spindle fibers disappear and nuclear membranes reform. And cytokinesis occurs in both cells. Meiosis II ends with 4 genetically different haploid daughter cells, each containing only 1 set of chromosomes. Some key points to remember about meiosis. It begins with a diploid cell. Meiosis only produces gametes. Gametes are genetically different haploid cells, sperm cells in males and eggs in females. Meiosis has 2 stages of cell division, called meiosis I and meiosis II. During meiosis I, homologous chromosomes separate to produce 2 haploid cells, each containing chromosomes in the form of paired sister chromatids. In meiosis II, the sister chromatids separate in both cells, becoming individual chromosomes. Cytokinesis of these cells produces 4 genetically different haploid gametes. And here are some key points to remember about prophase I. The pairing of homologous chromosomes, called synapsis, occurs. Each pair of homologous chromosomes, consisting of 4 chromatids, is called a tetrad. During the process of crossing over, chromosomes in homologous pairs exchange segments of alleles. Crossing over results in genetic differences in gametes. All gametes produced by meiosis are haploid.
All right, so this video was actually really descriptive, and the stages are very clear to see. So what I encourage you to do, replay it in your own time and also reflect on the stages from the notes. If you can split your screen and look at the description that is occurring in each stage, and also look at it in time in the actual visual that you saw, it will help cement everything that you have learned. What I would like you to do, now the link is here to the video. You just have to click on the link. It will direct you straight to the video. I want you to take down those key points to the very end of the video here. All right, so at timestamp 5:40, take down all of these points, and also take down at 6:21, at timestamp 6:21, take down these. What is synapsis? What's a tetrad? What is crossing over? Right? Synapsis is a fancy name for saying pairing of the homologous chromosomes. All of that takes place in prophase 1. Tetrad is when you have 4 sister chromatids of paired homologous chromosomes. Crossing over is when you have the segments of genes being exchanged between the homologous chromosomes. And then you will notice in the notes, in my notes, you would see a word called chiasmata here, which is simply the exchange of genes that occur on the chromosomes. So those are some terms that I want you to learn and be familiar with. So in case you have to describe what is taking place in each, you have those scientific terms to learn. All right, so any questions with these stages here? Anything you want me to go over? All right, so now we are going to look at a comparison between sexual and asexual. Why this is important in cell division is because we've learned that asexual forms the basis of asexual reproduction is mitosis, and the basis of sexual reproduction is meiosis, right? So that's why you would see this table in this slide. So with sexual reproduction, we've learned that it involves 2 parents, male and female. So you need to have sex cells called the gametes available in order for fertilization to take place. Fertilization is when you have the male gamete fusing with the female gamete, and it's the nucleus that has to fuse together so that the exchange of the chromosomes, the haploid number of chromosomes come together to form a diploid number, which forms an organism. There's the initial stage in the development of a human. Asexual, in the other hand, is 1 parent, no gametes, no fertilization takes place. So as you saw in all of those asexual methods, there's no mention of any male and female gametes or any fertilization taking place. Now, there are several advantages with both. For instance, with sexual, because the offsprings are going to have their own unique set of genes, they are different from each other to the parents, from each other and to their parents. They are different than their parent. It also, and that's a good thing, because sometimes if you have the same set of qualities, same set of traits, it can lead in, it can actually have very poor traits that will pass on, and your survival your survivability may be lowered, meaning if your parents they have let's say some genes that were of poor quality, then you yourself will have poor quality genes and you will not be able to survive or adapt to the environment as well as someone who has good quality genes. Another advantage is it produces variation in the offspring, so organisms gradually change. Once there are different genes, the offspring can, the fittest species will survive and they are able to make other species that are fit. So they can change and evolve. And lastly, there will always be a proportion of organisms which can survive and adapt to unfavorable changes, so the population is less likely to die. So all of these 3 points are very similar. All right, and then asexual advantages are offspring can be produced quickly. That is because it doesn't require a mate. It's only one parent, so there's no purpose for sourcing a mate in this case. And because it's only microorganisms or some plants that can produce through asexual means, they are smaller, so therefore they are able to be produced faster, all right, than a animal or a human. Then there's often large numbers of offspring. This is useful to parasites where any individual stands only a small chance of finding a new host. So because the reproduction process is very quick, you can have a larger amount of offspring being produced at a given time. And then another advantage is that desirable characteristics can be artificially bred into a population, giving them certain advantages. For example, farmers have been able to produce high yield or disease resistant crops and livestock. Now we have seen in artificial vegetative reproduction, we've seen grafting, we have seen tissue culture. Those are two artificial ways in which you are actually choosing what plants you want to reproduce. You are choosing desirable traits that you want, like the mangoes, what I mentioned. Remember, if someone wants big, juicy mangoes that are very attractive, they can actually choose which plants they want to obtain those traits from and breed a new plant, right? Form a new plant. Same thing with dogs. You see it in dogs where the owner may want a guard dog. You all know a pit bull was not naturally made. Pit bull was an artificial breeding process where they took American Terrier and they breeded with a bulldog. So those two, when they mated, they formed this what you call nowadays pit bull. So it have traits from the American Terrier, that ferociousness, and they have that bulldog look where they have a lot of muscles. They are very lean. All right, and the reason for having that breed of dog is to basically have that guard dog, have that dog that can offer security purposes and so on. So that's another advantage where people can actually choose little characteristics that they want in plants and animals to produce. Now, a disadvantage for both, we are seeing for sexual, mating partners must be available. So in order for reproduction to take place, both partners must be present, must be fertile, meaning that their gametes must be, healthy and fully developed. Also, there are fewer offspring that are produced. Reason being because it takes a while to find a mate. It takes a while for reproduction to take place. As a result, you have less offspring being produced at a given time. Like for instance, humans, reproduction or let's say pregnancy takes 9 months, right? That's way longer than if you were to have bacteria producing and reproducing in a lab. If you have bacteria growing in a lab, it takes a couple days and you have millions of bacteria. All right, so that's a difference in terms of how many offspring can be produced. So that's a disadvantage. Asexual have some disadvantages whereby offspring are genetically the same as their parents. So if you are a clone, and let's say your parent was not the strongest, and you are identical to the parent, then you have a less survival rate. You can get a disease easily. You can be wiped out if the environment changes very easily. Also, there's no variation, so population is less likely to survive adverse changes to their environment. Let's say the temperature has gotten really hot, and because you have one set of genes or you are a clone, you don't have that variation, that variety of alleles in your body, you are unlikely to survive because you could not adapt and you could not overcome that change in the environment. So we see those disadvantages taking place in asexual. And that is basically it for today's class. I would leave you all to go through that notes from the video, take it down. Also take down if you have to redraw or if you have to print, what have you. I encourage you to take down this table in slide 6. All right, and then study for your mitosis and meiosis quiz for next week. I postpone it so that you have enough time to really go through the two types of cell division so that you can understand it more clearly for next week. And after the quiz, we will start a new topic. So if you have any questions, feel free to ask. I'm going to close off.
Excuse me, miss, I was on to know if one will be doing cells.
Cells is one of the earlier topics, but we will, I want to finish genetics first, which is section C, that coming next. So I'm going into section C and then I will go back to those old topics when doing past papers.
Alright, miss.
Did you look at the recorded classes